Good morning, everyone. It's a pleasure to welcome you for this, I would say, special I3h seminar. We have the pleasure to welcome today Jean-Michel DOGNÉ. Most of you are aware that Jean-Michel is an important member of the vaccination task force of the Belgian government. But I would say that also when we decided to invite you, it was not in this capacity, but we were thinking that it's critical to understand how this pandemic is addressed, especially at the level of the development of vaccines, to have an international perspective. And obviously, this is what you will bring us. Jean-Michel, you are head of the Department of Pharmacy at the University of Namur, but also you are a member of the European Medicine Agency Pharmacovigilance Risk Assessment Committee. And as you know, vaccine safety is one major concern as we speak, I would say. But not only are you extremely active and you have an important role at the European level through the EMA, but you have also an even important role at the global level as member of the WHO Global Advisory Committee on Vaccine Safety. So we are very honored and pleased to welcome you. As we all know, the only available vaccines, so approved vaccines so far in our country are vaccines based on messenger RNA. Other vaccine will come later. Perhaps you will say a few words about this. But today we will focus on the safety of COVID-19 mRNA vaccines. And the title of your talk is indeed about what we know about their safety and how we can mitigate the risks associated with these vaccines. Jean-Michel, the floor is yours.
Thank you very much. I hope that you have my slide on the screen. Yes, can you confirm that?
Yeah,
yes.
I confirm.
So indeed, thank you. I will focus on the mRNA vaccine for two reasons. First, because these are the two ones that we have currently onto the EU market, but not only the EU market, but that also we had few information regarding the real use in real life data in terms of safety, but also in terms of efficacy. So I will directly focus on one slide, which seems a bit busy, but I guess that all of you know the concept of the SARS-CoV-2 virus to enter the cells and to create the pathology itself, which is the inflammatory phase with a prothrombotic state, and the difficulty still today that we have to find or to develop adequate treatment other than repurposing. So I mainly put the repurposing drugs on this slide, and you see that we have many repurposing drugs on the slide. And again, in the global context at the level of the European Medicine Agency, what is only and mainly recommended as marketing authorization that follow the same rolling review process and conditional marketing authorization is remdesivir, which is onto the EU market, but for which the WHO did not put a strong recommendation of using it, if not the otherwise. They do not recommend it to use remdesivir. So we are still left with a series of drugs that are currently under investigation, but that 300 are under current investigation. Some papers has also been recently published with the IL-6 modulator that seems to maybe providing interesting as well in the mortality associated to the severe cases of COVID-19, but still today what the European Medicine Agency and the WHO recommend is the use of dexamethasone or corticoids 6 milligrams once a day for 10 days, and to prevent the prothrombotic state with low molecular weight heparin and enoxaparin, mainly 40 milligrams a day. We have a series of talks by Michel on the antibodies that could be used also to treat SARS-CoV-2 disease and SARS-CoV-2 COVID-19 disease. Some of these are marketed by a urgent authorization at the FDA level, not yet at the EU level. And by the way, we do not have the same at EU level. We do not have the same possibility of urgent marketing authorization. But still, there we had also at the level of the EMEA now some discussion with antibodies targeting the S1 spike protein. So what are we left? We are left with using human also convalescent plasma in terms of antibodies, or of course the use of vaccines. So the vaccine, this was a lesson learned from also the past, especially for Ebola, that we need it, and it was, if not an obligation, it was in terms of public health. What was needed is to develop a vaccine in the shortest period of time. And you know that when it comes to a traditional development of vaccine, we have many limitations that are not only administrative limitations, but are the cost of the development of the vaccine and collaboration to come to a vaccine in a short period of time. And in the past, one of the fastest developed vaccines were in between 4 or 5 years. But you will see that more globally, it's much more than that. And so I would say what was needed here is to develop a vaccine in less than 1 year, especially when we have seen that repurposed drugs and other drugs into development did not provide a strong efficacy or effectiveness in terms of prevention of the disease or treatment even of the disease itself. So it led to the development of rapid vaccine by non-clinical data that were shortened or that were performed in parallel with clinical studies, and also clinical studies that were developed quite rapidly, and for some in parallel with different phase studies. But still the phase 3 was, for the different vaccine, one of the most powered studies to study the mRNA vaccine, but not only the mRNA vaccine. And at the level of the European Medicine Agency, we reviewed in a rolling review all the data for the different vaccines that have been asked to be reviewed like that. And there was also a possibility to have the production, the upscaling quite rapidly before the marketing authorization, both by the FDA and by the European Medicine Agency. And so now you know that we have the vaccine onto the market, but what are the differences already in the vaccine developed when compared to the vaccine developed for more than 10 years now? So there was a publication in JAMA late in November 2020 that showed the characteristics of the 21 vaccine approved by the FDA. And you see that we had 21 vaccine approved in 10 years, but the duration of follow-up for serious adverse event was usually 6 months. So that was a challenge, of course. If you want to develop a vaccine quite more rapidly, you need also to question yourself, do we need a 6-month follow-up in the clinical trials? And what was also quite interesting with these data is that the patient in the intervention group as a median was around 3,500, which means that it are not such power to detect adverse events. So even if you do longer study, which is such a power study, you will still have only the possibility usually to detect with 95% confidence a maximum of 1 out of 1,000 events in this population. So what usually we do not know is that these studies are usually not that much power to detect rare events with the vaccines. And the development of these vaccines were quite ambitious from different companies working together, or also in the efficacy and safety in Postmarketing. And we had viral vector vaccines that also emerged as a possibility with the replicating viral vector, for which we have some experience, and the one for which we have experience, at least in the exposure, are non-replicating viral vectors such adenovirus vaccine with the Janssen vaccine Ebola vaccine. Okay, so what was the new real challenge? And I can tell you that when discussing with the WHO and the EMEA back to March 2020, was that we were a bit surprised that many companies wanted to jump on the use of RNA vaccines for the development of COVID-19 vaccines for different reasons. But the first one was because of the lack of experience with these vaccines in real life, and even in clinical trials, huge clinical trials. But still, and you know now that at least 2 companies wanted to start with this RNA vaccine, Pfizer, Moderna, but also more locally as well with CureVac. And Pfizer had not a lot of knowledge in the field of this vaccine, so they themselves with BioNTech. And at the end of the day, it proved in terms at least of clinical trials that they could conduct quite rapid and efficient clinical trials. And you see that, interestingly, the majority are protein subunits for which, at least at EU level, we do not have this vaccine on the table of the European Medicines Agency, and the viral vector non-replicating being the second one. But you see that the RNA vaccine just represents around 10–11% of these vaccines under development. So only few wanted to, I would say, challenge themselves to believe that the RNA vaccines would be the best in terms both of efficacy and data on safety. And so I will focus on the development of the vaccine and the follow-up of the safety at EU level. And I put on the slide the different stakeholders in the vaccine development at the EU level. Of course, we have the vaccine developers, and at the EU level, we have 3 different stakeholders. First, the European Medicines Agency that I would say proposed the assessments, the evaluation of 3 different aspects: the quality, the safety, and the efficacy. And this is done by the scientific expert group, the committee, the CHMP, and the PRAC. But there was also a new committee that was created, or I would say a new task force that was created. You know, task force is quite fashionable wording to be used now. And they have created the EMEA Pandemic Task Force, where we present also the data by the vaccine developer and where we assess the data. And the CHMP provide an opinion to the European Commission that proposed a marketing authorization, a conditional marketing authorization in these cases, because we have few data or a series of data or studies that are still ongoing that will provide also important information. And at the end of the day, they only provide a marketing authorization in the different member states, 27 member states at EU level, for which they need to implement the use of the vaccine. So the vaccination rollout depends on the countries, and in Belgium, it depends on the different regions by themselves. So it's even more, a bit more complicated. So at the time of the mRNA vaccines discussed and developed, we had to roll out as well and to make the rolling assessment of the data coming from first, I would say, the quality of the vaccine, and then the efficacy and the safety coming from non-clinical and clinical data. And now we are still working in parallel to work on the quality, efficacy, and safety data from still today, non-clinical data, clinical data, but also post-marketing. And so we had to learn a series of lessons from a novel vaccine that were introduced in the past during pandemic. And the main was of course, the 2009 H1N1 influenza. And we knew that in some countries, and this is at the WHO level, so at the worldwide level, that a few countries were prepared for pandemic vaccines use and good vaccine use in their population. On top of that, we knew that we might have some theoretical risk for which we needed to investigate what would be the impact of this vaccine, and what was of the major fear was the vaccine-associated enhanced disease. Because as we know, the concerns that we have with the pulmonary events associated with the disease itself, what we didn't want is to have a vaccine that may create an enhance of the disease if the vaccine is not adequately developed. And on top of that, we might have the concern with the adverse event that may arise after the vaccination, for which we would not have anticipated that they may arrive, and for which we do not have the background incidence rate of the data. And this is mainly true for the 2 mRNA vaccines and non-replicating vector vaccines, for which you see that if they have or they might have advantages, one of the background, one of the difficulties, that we have with the lack of backgrounds with this vaccine is the lack of experience in safety for both the non-replicating vectors and the mRNA vaccines. So it was a huge challenge, not only in terms of efficacy, but we expected to have good efficacy, at least from a phase 1 and immunogenicity study. but from clinical studies, both because of the disease was quite difficult to investigate, but also in terms of safety, the benefit-risk balance was quite challenging to anticipate. And what was, as I have mentioned, needed to anticipate is the possibility of to have a vaccine-associated enhanced disease, and more specifically, a vaccine-associated enhanced respiratory disease. And so we might have two different syndromes that we needed to, I would say, anticipate. The antibody-dependent enhancement, which was seen at least in vitro with SARS-CoV-1 vaccines development, but also the vaccine-associated enhanced respiratory disease, that is a condition which is associated with non-neutralizing antibodies that can lead to complement activation for some specific cytokine production. And this is interestingly based on the Th2 response. So there was a need to address this not only non-clinical study, but also in clinical studies and in post-marketing safety. And we had some cases in the past with some vaccine that did not produce such good neutralizing antibodies that could lead to such enhancement of the disease. So it would be, of course, a real challenge. And so from a clinical safety point of view, we had some guidance at the EMA level to consider before conducting these clinical trials, but not only at the EMA level, but also at the FDA. And we were a bit, I would say, not on the same page for all, but what we knew is that most adverse reaction to the vaccine occur within 4 to 6 weeks after vaccination. So I told you that usually we want to have data with a 6-month follow-up of vaccination to get a marketing authorization. But in this case, we anticipated that since the majority of the adverse reaction occurs within 6 weeks, we would have accepted the data, at least interim data, that would come with a median of the population treated for the 2 doses in the clinical trials with 6 weeks follow-up. The FDA asked for 8 weeks follow-up. And secondly, we wanted also to have recommendation that the clinical trial would continue for at least 1 year after completion of the vaccination, and usually it is more than 1 year with now the different vaccine under development. This is up to 2 years. So I would say the vaccine developer follow this. And I put on the screen where we get data from the vaccine in general and the mRNA vaccine. So again, the data that we want to have is good quality, of course, of the vaccine itself, because lack of good quality may lead also to impurities that may lead to some toxicities. And we know that with the mRNA vaccine and the upscaling with this vaccine may lead to impurities. So we needed to have some guarantee on the quality of the vaccines that mRNA produced was of good quality. And there we had some discussion, and we are still having additional data that come with the quality development of these vaccines. But up to now, so far so good, the quality is in line with the current standards that led to the marketing authorizations. You will see that there are non-clinical data that were needed, especially for the enhanced potential disease with the vaccines and the clinical trial. And one of my main topic is, one, we have the decision making. What do we do in the post-marketing setting? So I will address these to impact to identify what could be the impact on the safety of the mRNA vaccine. So first, as I have told you, what came from non-clinical data, we have usually good non-clinical programs, of course, with all medicines that follow I3h guidelines. So guidelines not only at national or European level, but international level more wide. And we had these non-clinical programs that were conducted quite adequately. And then we see from an efficacy point of view, but you all know that the mRNA vaccine, but still we did not expect that without the data, that they produce not only neutralizing antibody, but they also produce a good cellular immune response to the spike antigen. But what we needed to have also was the information about the type of immunogenicity at cellular level that it would develop. And you see that for both these mRNA vaccine, it induced a type 1 helper T cell, so a Th1 response, and lower undetectable Th2 response, which was quite interesting since it is considered, of course, not really validated, but as one of the most important biomarker of a risk of enhanced disease. And in the classical convention studies of the repeated dose, for instance, toxicity with the vaccine, they did not they did not reveal some special results. So what does it mean is that it led to the possibility to conduct these clinical trials, but still, even after these clinical trials, today we have still some non-clinical studies that are ongoing. And what I have told you with the power of these studies is that everything that you want to find in the clinical trials in terms of safety depends on only one thing: the diversity of the population included in the clinical trials, and of course the power of the clinical trial. So the less people you have with the less diversity, the less even adverse event you will see in these clinical trials. And there I would say not by chance, but because it was driven by the power needed to conduct good efficacy study. The only possibility to have good efficacy study was to have enrollment of people that would allow to find some events associated with efficacy. So what does it mean? It means that you needed to have usually at least 15,000 people treated with the vaccine compared to a comparator, to have some cases of COVID-19 vaccine for which you can identify if it is different in between the two groups. So you needed to have high-power study. And when I say high-power study in terms of safety, this is the goal that we want to reach. And this is where usually the company do not want to have so many people involved, because usually it costs more. You have data on efficacy that is enough, but you do not want to have more people in this study because it would take too long time, it would cost too much, and it may lead to identification of adverse event. But in this case, you see that we have high power studied, much more power than the majority of the vaccine. What does it mean? It means that we had, with 15,000 people treated in the vaccine group, we had only, I would say, few chance, 22% of chance of missing rare side events in these clinical trials. While with the common or classical vaccine clinical studies, around 2,500 or even 5,000, you have much more chance to, I would say, miss rare side effects. So this was quite important that we had some data, and you will see that we had some data on rare side events that we put in the SMPC of this vaccine that we would not have ever seen in other studies. And the second point about the interest of having the study of adverse event in this population is that I have said that you need two things. First, to be highly powered, and secondly, to have few excluded people. And indeed, in these clinical trials, you have few excluded people. So you had good data with different group of ages, including the over 65 and mainly the over 75, where we had still 4.3%. So it was a bit limited in terms of variability, in terms of estimate about the efficacy, but again, in terms of safety, having these people, it is usually not seen in clinical trials with the vaccine. We have few cases usually with clinical trials with vaccines over 65 of age. So we had good, interesting data there. And we had also good, interesting data in comorbidities. In this case, with the Pfizer vaccine, we had around 46% that had some comorbidities, mainly obesity, but the different comorbidities were quite good represented, including diabetes, for example, and chronic pulmonary disease. Usually these are excluded from clinical trials, or at least I would say not really represented in these clinical trials. But you see that here we had more than 100,000 of people included. For some of these, they represent more than what we have with usual vaccines. For the flu vaccine clinical trials, it is around 1,000. So in this case, in some specific population, we have more cases and more people involved that we have for some clinical trials as well. So, and you see for hypertension, for instance, we had many cases, of course. So usually with the vaccines in development, we have the adverse event that goes up to, I would say, common and in some cases uncommon. And so we see in these studies that we can indeed view that the immunogenicity is associated with a classical reactogenicity, with headaches, with myalgia. Pyrexia was not that much elevated. It's mainly elevated, and I will come back to that after the second dose, not after the first dose. And more generally, this is usually more after the second dose that we have these events than after the first dose. But you see that you can go to uncommon events that were clearly seen in these clinical trials. We have a series of cases of lymphadenopathy, which is currently associated, of course, with an immunogenicity. But what is interesting, and you see there, this was a lot of debate, and we also debated with that on the phone with Michel, with Bell's palsy, so acute peripheral facial paralysis. We have seen some cases, and the two different studies, the one from Pfizer and Moderna. And what was a bit strange is that usually the background incidence rate of Bell's palsy is 1 out of 10,000. And so we would expect to have at least some cases in the clinical trial with the vaccine group, but also with the placebo. And what was a bit strange is that we had no cases of Bell's palsy in the placebo group, with more than 20,000 individuals in the placebo group. And we had 4 cases with the vaccine treated. But these 4 cases were, the onset was a bit different. We had 37 days after the dose 1. And this participant did not receive the second dose. And for the other one, it was mainly after the second dose, but with timetables that were quite broad, 3, 9, and 48. So only, I would say, 4 cases, which was expected. A bit less was expected than 4 out of, I would say, 20,000. We would have expected maybe 2 out of 20,000, but interestingly, there was no case in the placebo group. So what we put there is that, yes, we had some cases reported. The causality cannot be clearly established because the cases are few. The concern was mainly that we had not cases in the placebo. And for the Moderna vaccine, it is 3 versus 1. And I would say this is more in line with what we would expect as a background incidence rate. But if you, I would say, make not a meta-analysis, but accumulation of the case, out of 70,000 in clinical trials with both Pfizer and Moderna, we would have 7 versus 1, which make a signals. And so this is the reason why we inform at EU level that it could be associated to the vaccine, but the causality is unclear. Why at the level of the FDA, they did not recognize that it could be causally associated, or at least not excluded. So if you go in the FDA and the Canada, they do not propose this statement in the information of the product. So this is interesting because this is what you can see in clinical trials for which you have more than 10,000 people, what you have not seen that with less than 10,000 people. So the data from the clinical trials were quite interesting in terms of safety and efficacy. Of course, we are not discussing the efficacy in this case, but what we needed to set up is also post-marketing vaccinovigilance. And why? Because it also impacts the benefit-risk balance once the vaccine is onto the market. And we have different tools to do that. The passive surveillance, I will show you how it helped now to update the risk management plan and the benefit risk of the vaccines. But we have in place also a series of tools, of which some I will discuss with you. You may know the spontaneous or solicited reporting, and what you may not know is that patients since July 2012 have the possibility themselves or the vaccinee, which are not patients in all cases, to report even to the national competent authority at national level. At Belgium level, this is the Federal Agency for Medicine and Health Products. But usually we prefer to have cases reported by healthcare professionals because usually there are more details. And there we have set up a system for reporting the adverse events associated with the COVID-19 vaccines that are more specific. So if you go on the website of the Federal Agency of Medicine and Health Products, on the patient cards, on the vaccines, Comirnaty vaccine, there is reference to report the events to this database. There we make causality assessment of each cases, and we provide these cases to 2 different databases, the Ultravigilance, which is the EU database, but also the Uppsala Monitoring Center, which is the World Health Organization database. And what's quite interesting, of course, is that even if we receive low quantities of vaccine in our population, even if we take some more time to have data about this vaccine at national level, what is interesting with the different database for where we provide the cases is that at least at the WHO level, we have more than 140 countries that participate to this program. And so we had, so the program, it is located in Sweden, in Uppsala, but it also depends from the WHO. We had the adverse event reported up to now with all the vaccines that are marketed in all the participating countries. And I have said there are more than 142. So even if the Pfizer vaccine was marketed first in UK, was marketed in other countries, including US, including Canada, and yeah, if we are a bit late when compared to them, this is interesting in terms of knowledge of post-marketing with this vaccine in other countries for which we can access to database and inform about the adverse event. And interestingly, what you see is that usually the more reporting countries in this database are also the countries that use now the vaccine the most. And you see here we have the US that is now up to today, it's around 14 million doses of the vaccine, the Pfizer and the Moderna, but mainly Pfizer, for which we have now good information in the different database regarding the adverse event. Of course, I put also Russia and so on, but they do not use the same vaccine, but still we can compare and we can do some disproportionality analysis with the different vaccine when compared to other vaccine in the different database. And this is good as well, because as you know, in the clinical trials, we have no, and I guess we will not have in the future, comparative clinical trials with the different vaccine, for instance, Moderna and Pfizer in the same clinical trials. You will not have that, especially for safety reasons. And what it can bring as an information, only the solicited report or the spontaneous report, is the vaccine reaction, especially the one that occur quite rapidly after the vaccination, but we have also some cases reported after a delayed reaction. And this is quite important, and this usually comes from not the spontaneous reporting, but what we call the active surveillance post-authorization safety study. So as in clinical trials, and Belgium is participating to that, we follow still in real life cohort of vaccinated people on a longer period of time so that we can have not only the immediate reaction, but also some of the delayed reactions. And why I did put this slide is that because this was published in New England Journal of Medicine in line with some cases that were reported with anaphylaxis reaction that were not seen in clinical trials. And why they were not seen in clinical trials? Because on the contrary to Bell's palsy, for which the background incidence rate is 1 out of 10,000, anaphylaxis, even associated to a vaccine, with the flu vaccine occur in 1 out of 1 million cases. So it's obvious that we would not have seen them in clinical trials. But coming from the cases that we had reported in this case at the US level, in the VARS database, we have seen that we have more frequently reported cases of anaphylaxis when compared to the 1 out of 1 million cases reported with the flu vaccines. Indeed, there was 11 cases per million doses, so it makes 1 out of 1,000 the frequency of these anaphylaxis. And what was quite interesting that comes also from the spontaneous reporting is that usually they occur before 15 minutes, and they can occur both in patient or treated people with previous history of allergic reaction. This is mainly the case, but also in non-previous history of allergic reaction. And when you look into indeed the cases more in details, you see that the median age was 40 years of age. And because, of course, not in Belgium, we have seen that much also, because usually the age was more median age, and we do not vaccinate these people again, still, I would say, in Belgium. And for those who do not know, the majority of the anaphylaxis occur with more than 90% in women. So when compared, of course, to male, and the documented history of allergic reaction was in 81% of the cases. So the majority with history of allergic reaction but not all cases. And so the causal association was needed to be assessed, and this is what we do when we do signal assessment based on cases, is to try to find a underlying mechanism. And what we know is that these mRNA vaccines indeed are not stable as such, so they need to be put in lipid nanoparticles, and for these we have difference in terms of composition between the Moderna and the Pfizer vaccine, but you see that we have 4 different lipids, and 2 are major ones. The first that is close to the mRNA, which is a cationic lipid, which is a specific one that was not that much used in the past, and a PEG lipid that allows to have a better half-life of the virus, of the vaccine itself. But as a comparison, when we had the cases, there are some cases, and the majority of the cases were coming from Comirnaty because it was first marketed, but we are starting to have accumulating evidence that we have with Moderna the cases. And in the 2 cases, they use different lipid formulation with no adjuvant and no conservative, but still one of the 4 lipids and one of the 2 main lipids at the outside layer of the lipid nanoparticle is indeed with PEG 2000. And we do not exclude that this PEG 2000 may be the reason for an increased risk of anaphylaxis reaction when compared to, I would say, the background incidence rates with other vaccines. But now what is quite important in terms of communication is always also to put into context these information with what is currently observed with other drugs, including other vaccines. I have told you that with flu vaccine more in general, they have one of 1 million cases reported. But if you take this in the database, the frequency of anaphylaxis reported associated to drug-induced events, you have around 1% that are reported. And the main common, as you surely know, is penicillin, sulfonamide, and SAIDs. But what's quite interesting as well is that we have some patterns of these anaphylaxis that may help to identify, for instance, people for which we may minimize the risk. And for instance, we have again, female sex may be the one at risk of higher risk of anaphylaxis. White race was also the case when compared to Black people. And you see that for some, we have some increased risk in the general population. For instance, asthma may be also at higher risk of anaphylaxis itself. And so what does it say is in this paper is that usually we do not do after these cases tryptase testing, and this is not recommended indeed for this vaccine as well, but the importance of educating the providers on of anaphylaxis management is quite a major one. And this is what we did at the level of the European Medicine Agency and also at the Belgium level. For 2 reasons. First, because we understood, and coming from UK first, that there was no case of anaphylaxis in clinical trials, but at the level of the UK, quite rapidly, the 2 days after the first, I would say, start of the vaccination, they had some cases of anaphylaxis reported. And we still had the dossier on the table at the level of the European Medicine Agency. So the MHRA at the UK level reacted quite rapidly, suggesting that they needed to contraindicate people that have history of allergy in the past, and because they didn't know how to handle these cases of anaphylaxis, were they more frequent or less frequent, were they appear more frequently, they didn't know. But back to now 2 weeks of experience before making the opinion at the level of the European Medicine Agency, we have seen that again, the majority of the cases appeared quite still rarely, but mainly before 15 minutes. And so this is why we propose that we need to have a close observation for at least 15 minutes following the vaccination, and that if after first dose of the vaccine you have some cases or you experience anaphylaxis, the second dose should not be given. And on top of that, within the vaccination centers, we have SOPs in place for which we propose to have also the vaccination under medical supervision with the material to be used to treat this anaphylaxis. And this is what has been used in the different centers where today we have the vaccine used. I will not go into detail with that because we have not published that, and we will propose that this week to be used as a recommendation. We have developed that with Pierre Van Damme and with specialized in immunology and immunoallergic reaction, to propose a quite easy, I would say, template to know what we need to do if we should vaccinate people with some history of some disease, with precaution, what type of precaution, with the follow-up not for at least 15 minutes, but for at least 30 minutes, and with some specific condition, for instance, hereditary angioedema, idiopathic anaphylaxis, how to consider vaccination of these people. And so this is based on some recommendation coming from the EMEA and from the CDC as well, not only for the first dose, but also for the second dose. And this will be released this week. And so the main difficulty that we have with these cases is to put them into the context of what we may expect to have as a disease in the population. And so this is some analysis that we do, I would not say every day, but quite frequently at EU level, which are called the observed versus expected analysis. The observed versus expected analysis is that you need to have the reported cases by the population, and you need to put in the context of the expected cases, so the incidence of the cases in the population. So we need to have background incidence rate of many diseases stratified by age, of course, and by geographic location. And we will need to see if at some point the frequency of the observed even in the population of this group of age comes to be higher than what was expected or not. So it's an important tool for rapid signal detection. It does not provide any information about the causality, but if you do that, I would say weekly or biweekly, then you can see a dynamic change in the observed cases reported, for instance, anaphylaxis, when compared to what is expected. And so it can be used also for communication. Of course, the main difficulties that we have with this kind of analysis is the uncertainties, because the observed cases is based on the reporting of the event both spontaneously and I would say activate reporting events. We need to know exactly the vaccine exposure to have the denominators of the observed cases. And this is why at the level of the EMEA and the WHO, we have stimulated the member state to have a registries, a traceability of the number of people vaccinated with which vaccine, which dose, and which brand name, and which lot number. If we do not have that, it's quite difficult to make good observed estimates, so the incidence of reported cases versus the number of people vaccinated. And so this was the main reason why we wanted to have registries at different national levels, so not at all for the traceability of the people that have been vaccinated. And the expected analysis, we need to have good background incidence rate before the vaccination starts. And this is where we made a huge job at the level of the WHO and the European Medicine Agency. And to put that into context, this is an example that we use when we had the HPV, so Gardasil and Cervarix vaccination, because we knew that we would vaccinate new population in between, I would say, 9 and 18 years of age women. And we knew that after vaccination, we would have a series of events for which we needed to know as well the background incidence rate. And to illustrate that, we knew that once we vaccinate 1 million of young girls, women, with a placebo, so without vaccines, how many cases of autoimmune disease would we have after this vaccination, with theoretical vaccination, since we do not vaccinate anything out of 1 million young girls. And you see that after 1 day, we know in the US that we have out of 1 million young girls or women, 4 cases of diabetes that are diagnosed, 1 case of thyroiditis, 27 cases of asthma. So let's suppose now that you change here the placebo by a vaccine. It could be, for example, for instance, the SARS-CoV-2 vaccine. then automatically you will have these kind of estimates which are the background incidence rate. And if you are in the media and then you say, yes, there was one case of diabetes that was diagnosed the day after the vaccination, so the diabetes is associated with the vaccine and the vaccine is the causal association. And you see that if you go 1 week after, we had, if you take one classical example, the thyroiditis that we suspected to be involved also with the HPV vaccine, we had 9 cases out of 1 million. So this is why we need to put into context. And what we did as a job and at the level of the EMEA and the WHO, we put a list of what we call an adverse event of special interest associated to the vaccines and most, whatever, I would say, the mRNA vaccine, but also the other ones. And we propose a series of events that may be associated to the vaccination. And to be clear, and I know that there are journalists in the room, this is not adverse event for which we know that the vaccine will create. This is events associated with the vaccine that we need to assess. It's quite important because I have seen in the media that this has been published, that this is what the vaccine may cause. This is not what the vaccine may cause. This is what we need to study after any vaccination as a background incidence rate to see if the events reported with the vaccine is higher than what is expected. And why did you use a series of events? Because we know that there are some rationale to use these events. GBS, Guillain-Barré syndrome, was in the past associated with some vaccine. There are also other autoimmune diseases. You know that we have anaphylaxis there. We know that it can be associated, but the frequency is needed to be clearly determined as well. And we have some adverse events that may be associated with some platforms. Of course, we do not have mRNA vaccine in this case, since there was no example in real life of this vaccine in the past. And on top of that, the list was also did also include a series of events associated to the disease itself. So I told you with the enhanced disease following immunization that were seen in the past in model with SARS-CoV vaccine, with the dengue vaccine, for which now we vaccinate mainly the seropositive people and not the seronegative people because it may create enhanced disease. For all these, and the multiple system inflammatory syndrome in children is a really specific disease now associated with the COVID-19, for which we wanted to have good estimate of the background incidence rate to see if in the vaccinated population, we would have, when reported these cases, an excess of cases when compared to the background incidence rate. And so we did that up to now, and we have this data for which now, even for the whatever the events associated to this adverse event following immunization, we can put into context. And another example to put that into context is, and it dates from last Friday, we had that in Belgium as well. You know that Norway investigated 23 deaths in frail elderly patients after vaccination. And it did, by definition, if you vaccinate frail patients, you have a risk of death because the vaccine does or may only protect or decrease the risk of having symptoms associated to the disease, but it does not protect death per se with the other disease. So you will not stop people in frail or very frail patient to die from the underlying disease with the vaccine. But the question is there an association, a causal association in between a vaccine and the development of or an increased risk of their own disease? And so that's a bit challenging, of course. And so the head of the Norwegian Medicine Agency told that today, there is no clear connection in between the death and the vaccine. This is currently under investigation, especially for 13 of the deaths. But what he says as well, for which we did not have at the European Medicine Agency level a clear-cut conclusion as he has, is that maybe some events, and I told you that the events such as fever and diarrhea and nausea may have contributed to the fatal outcome in the frail population. That's a bit of a challenge statement since we do not have that, in fact. And so again, we can recommend, and there are some recommendations in the BMG journal that comes, that still is in line to us with how we use the drug, how we use the vaccine. This is a vaccine that needs to be used under medical supervision as well, and for which there is no obligation to vaccinate a very frail population. And for the flu vaccine, for which we vaccine also the over 65 with frail condition, it's also up to the physician to decide if he is fit or not for the vaccination, with that includes the even associated with the vaccination after the first dose and after the second dose. And to put that into context, and we make also the estimates at Belgium level, where we had some cases, of course, of deaths after the vaccination. In Norway, there are around 400 people that die in care home resident every week. So every day there are around, I would say, 50 to 60 people that die outside of the vaccination. So this is a way, I would say, also to put that into context, but we need to investigate all these cases and find the best way to follow the frail and the frail patient. And we had some information that we know that there is no dose adjustment with the mRNA vaccine in this population, but still the vaccination should be postponed in the individuals suffer from acute severe febrile illness or acute infections. But we do not have clear statement with frail or very frail patient, because there are many definitions of this I would say in the literature, but we did not provide any recommendation in frail or very frail patient. But it's still a matter of benefit-risk judgment for vaccination of this population. And so this is what we know, but there's also many things that we do not know, and only few coming from the clinical trials, because they mainly included all, I would say, possible participants, but still not all: children, immunocompromised patients, and pregnant and breastfeeding women. And this is the aim of the risk management plan. The aim of the risk management plan that is proposed by the marketing authorization holders or by the applicant and the agencies are to identify risks that may or that need to be addressed post-marketing, and for which usually we have few or little information coming from non-clinical and clinical trials. And I put in this table the two ones, Pfizer and Moderna, and you see that we have identified the anaphylaxis as a risk, and we have risk minimization, as I have told you, in the how we use the drugs. What we have as an important potential with, because it remains theoretical, because we did not see anything in non-clinical data and clinical data, is the enhanced disease associated with the vaccines. And so this needs to be studied in the post-marketing, in the real life, not only from post-authorization safety study, but also from reporting. And we have a series of missing information. And you see that we have pregnancy, immunocompromised patient, we have frail patient with comorbidities. Indeed, we had data, but it's missing information in terms of large cohorts. So this is studied in real life as well. And the example of this that occur after the vaccination are part of the program. And the long-term safety data, of course, as well, and the interaction with other vaccines, we do not know. What we said when we said we do not know is it means that we need to know. It means that there is plans, and this is the aim of the risk management plan, to address this question with post-authorization studies or with risk minimization, additional risk minimization. And one of the additional risk minimization was the importance of the traceability. I told you why, because we need to know how many people have the vaccine, which brand, which lot number, and which dose. And this is why we propose also to have traceability and vaccination reminder cards. The vaccination reminder card is also there to remind of the second dose, when the second dose is needed, and to remind about events that may be associated to the vaccination that need to be reported to the federal agency or national competent authorities. And this is the aim of the traceability, not at all the traceability to follow people who has been vaccinated with what in the future. And I put just into, for your information, and I did not go into detail with what we call the pharmacovigilance studies that are ongoing with these two vaccines. in the different population for which we have missing information. For instance, immunocompromised patient, we have often the question, can we vaccinate? So we have informed that yes, we can vaccinate. We do not know about the efficacy in terms of immunogenicity, which expected to be lower than in normal population. But there are a series of clinical studies proposed, or a series of post-authorization studies proposed by the two marketing authorization holders. We have at least one interventional clinical trials with a Pfizer. In fact, it's two clinical trials, one in EU and one in US with the Pfizer vaccines. And we have observational studies of the outcome of this patient and the safety of this patient in EU and US database, only for the use in immunocompromised patient. For the Moderna vaccine, this is one observational and again, one interventional study. So we will have data on this in the near future, but we do not have. And usually when I get the question, I answer, yes, you can be vaccinated, or you can wait. And we wait for what? Wait for having this data, and we have timelines for this data, for these studies that are addressed in the risk management plan. If you want to have them, of course, it is open for transparency. They have the timelines with some of the protocols. The same for pregnancy and breastfeeding. We have for the different pharmaceutical industries some obligation, some commitment to do studies, not only observational studies, but for some interventional studies. So we are gaining more and more information every day with these two vaccines in the real life, mainly coming from registries. And at national level, we have also an international level registries that follow the use of this vaccine in pregnant women. The same is true for autoimmune and inflammatory disorders. Both Pfizer and Moderna will conduct observational studies, but there are also interventional studies. For instance, for the Pfizer vaccine, phase 2 in a patient with rheumatoid arthritis treated with immunomodulators with rheumatoid arthritis with 100 patients. Of course, with such a power, it will not provide data about the clinical efficacy, but it will provide data about immunogenicity. And Arnaud Marchand mentioned also that at the level of the Belgium, we are also conducting clinical trials with Sanofi in the coming weeks with some population, with some, I would say, specific population at national level, especially, for instance, cancer patients and also kidney disease patients. So I will finish by saying that at the level of the European Medicine Agency, they have driven these vaccine monitoring preparedness plans with lessons learned from the H1N1 pandemic back to 2009, but they do the signal detection with really some specific signal detection method using their own database that with all the data coming from the 27 member states, which is much more powerful than what we can do at national level, but this is generated by data coming from national level. And they have also proposed the access program, which is the program that proposed us today to have the background incidence rate, which allow the observed versus expected analysis. We did that before having the vaccine onto the market. So now we are prepared with every event to put that into context with the background incidence rate. And we have also pregnancy studies, I would say, registry or in line with, on top, I would say, of what the companies and what the pharmaceutical company do. So I also say that for the journalists also that say that usually only the pharmaceutical industry is responsible for doing the studies, providing the information, and we only rely on that to make our decision making. This is not true. We also focus on our own data coming from our own studies at EU level. And why is it important? Because usually when we know, and this is at the level of the Belgium, but also at EU level, the race is more why we did not vaccinate so much and so rapidly, why 2 or 3 weeks ago is that a few wanted to be vaccinated because the vaccine was developed too rapidly. But so now we have a good vaccine coverage, and we know that we are starting to get adverse events, real or perceived, that may indeed lead to a loss of confidence with the vaccination campaign. And I can tell you that if the vaccine is as efficacious as it is in clinical trials, we might expect in the few weeks or in the coming months an impact of the incidence of the disease. And if we lost confidence in the vaccine based on, I would say, even mentioned in the media, which are not associated, causally associated, it may has an impact on the coverage of the vaccine, which usually is associated with a new outbreak of the disease. And this is usually what we have with the different vaccines. So, of course, we need to be aware of the vaccine euphoria and beware of the vaccine euphoria, because we need to address all events reported, and we address all events reported. We put into context all these events. For matter of transparency, we put all the data of these signals on the website. The Belgium Federal Agency published every week all cases received. At the EU level, we will publish the first report. You have seen that the CDC published a report with the anaphylaxis. So, but this is associated with usual vaccine development, as with any drugs. With any drugs, we might have events, and this is put into the context of the benefits of the vaccine and the benefit-risk balance. And this is why I think this is not the beginning of the end, but the end of the beginning of the COVID-19, I would say, story, since now we have, on top of the treatment and the development with the vaccines, many things to resolve, many things to develop, many things to use, and many things to accept as well, because no medicine is without risk. They might have some risk associated with the vaccine. The context is important, put in the context of the benefit of the vaccines, and there will still be also quite important information. And I want also to tell you that we had published, and I have contributed to the WHO safety surveillance manual of the COVID-19 vaccine. We have published that some weeks ago. This is available freely online. This is more than 200 pages, and it also provides this kind of information in terms of communication, contextualization, adverse event of special interest worldwide. So if you are more interested to have details on this, you can download it, or you can read it in the PDF. Thank you for your attention.
So thank you very much, Jean-Michel. I think that was a very comprehensive talk. I think it was important to go into some details and explanation as you did. So now we have time for questions, just for everyone to know that we will close this meeting at 2 at the very latest. So now I will take the privilege of being the moderator to ask you the first question. As you know, there are some similarities between this coronavirus and the respiratory syncytial virus, especially obviously those are two viruses which are extremely are inducing important pulmonary damages. Obviously, the major difference is that young infants are vulnerable to RSV and look more or less resistant to COVID-19. But still, I asked the question regarding the RSV vaccine, which, as you know, has to be withdrawn for the market. This was, I know, several years ago. But my question is, do you think that nowadays we could avoid indeed to include or to allow the marketing of such a virus, and would it be prevented in the current regulatory environment?
I think that's really a tough one, because of course, as I have said, even for the enhanced disease, we do not have two things. First, a good biomarker for the enhanced disease, and the Th1 versus Th2 is one of the most recognized, but not the absolute, I would say, biomarker of the risk of enhanced disease. And secondly, we did not even have a definition of what is a vaccine enhanced disease. So we have, with the Brighton collaboration, proposed a definition on that. And this remains theoretical, the risk is very low, and in clinical trials, even with these clinical trials with 30,000 people, we would not have seen something, and we did not see something. So while the question is tough, is that I think that maybe today, and on top of that, with again this vaccine, we did not have good models of enhanced disease, animal models of the disease itself, and enhanced disease on top. So now we're starting to have this kind of model. So usually what we do is first we expect to have animal models that will help to first answer the questions, and what we did not have at the time of the RSV vaccines. So they were developed with classical tox models, which were not power to detect the events that we have with the RSV vaccine once marketed, and the clinical trials were not power as well to show that. So I guess that today, with the experience that we have with the vaccine, as we did with rotavirus, if you remember well, RotaShield was withdrawn from the market, and Rotarix and Rotatex were put on the market with higher power studies, with strong post-authorization safety study that still identify into susceptions, but with a, I would say, low extent that we had with RotaShield. So I think that today we would be more prepared to have additional non-clinical and clinical data to have this vaccine onto the market. And I think that maybe we would have the same as we have with Rotarix and Rotatex when compared to RotaShield, vaccines that will not lead to such an increase of the risk, but that will be more studied to prevent high number of cases in real life that would be expected. So today, I think yes, but back then, no.
Okay, thank you very much, Jean-Michel. Now we will move to other questions. I see several of them. Before opening the floor to the question, I think it's important to mention that Jean-Michel DOGNÉ has to declare it as no conflict interest at all in his different function. And actually, I think it's important to indicate that experts like Jean-Michel appointed by EMA or WHO, and I think that either your declaration of interest is probably made public. I think that's very interesting. It's very important to mention this. Having said that, the floor is now open for questions, and perhaps we will start with Elie Kogan, who was the first to ask the question. And for sake of time, because I see several questions, please focus on the question, avoid your own comments and go straight. And Jean-Michel, please answer briefly.
The question by Elie Michel was actually answered after the question was raised. It was about the cases, well, the deaths in Norway. So we will directly go to Jesse Thomas from KU Leuven, who has a question.
I was wondering what we can still expect from the long-term safety in phase 3 studies, when we know that a lot of the placebo, the participants who receive placebo will now receive one of the licensed products in the coming weeks or months. So what can we still expect from these thousands of patients or participants who receive placebo?
That's an excellent question. I think that we have discussed that back and forth at the level of the EMA, WHO, but also the FDA, is it from a medical point of view still acceptable to go on with these studies when you see the data about the efficacy of the vaccine when compared to placebo, and to have people exposed with the disease and keep going on the placebo? And it was concluded, and the EMA published, I think the FDA published, the WA published on that still today, we need to, as much as possible, continue with the placebo control follow-up of these studies for at least 2 years. Of course, from an ethical point of view, we cannot stop people from dropping the study and asking to be vaccinated with a more active vaccines. And so we will see what we will have at the end of the day, but we anticipate to have much less placebo that we have today. But still, as I have said, we can compare to placebo, but we can compare the vaccines and the risk associated to the vaccine itself to the same population which would not be vaccinated. So the background incidence rate of the disease, for instance, I put the instance of Bell's palsy. Even without placebo, I can tell you that what is expected with Bell's palsy in the same population when compared to the vaccinated. So we have tools also to assess the incidence of events, even long-term events, in only a single population vaccination when compared to a background incidence rate at the same time in the same population or back to historical controls. Of course, we would prefer to have it in the clinical trials, but we have tools to do that. We need to do that, and we need to still maintain the placebo-controlled studies. The more difficult the difficulties are for the next vaccine that are currently or will be developed in phase 3. Yep. Do we still recommend control with a placebo or non-inferiority studies with the comparators? And there we have also some difficulties, but it can still be produced in 2 ways.
Okay, thank you.
Okay, I think there is a question from Yves Carlier.
Yes.
Please, Yves, go ahead.
Yeah, thank you very much. Have we enough back in time to be sure that antibody-dependent enhancements will not occur by vaccinating previously infected patients in case of new contact with the virus?
We had, as you mentioned, back in time, so I guess that you mean the vaccination of seropositive people to have an disease. Okay.
What experience we have on that, yes.
Okay, the experience is limited because we had on the top of my head with the Pfizer vaccine, we have around 500 people that were seropositive that were included in the studies, for which we do not have data on efficacy, but on safety. On the safety side, they had some specific concerns if they were seropositive when compared to all the major group of the seronegative. The same being true for Moderna. But of course, this is under power to identify, for instance, the enhanced disease, which we are quite convinced this remains very theoretical, and we would have seen it as well in the seronegative people in clinical trials, and we did not see it. In the post-marketing, we are looking for it. We do not see today an enhance of the disease, but what I can tell you is that from my personal point of view, is that if we change the dosing that we have in clinical trials, if we do not use 2 doses anymore, but only a single dose in naive patient, even in seronegative patient, is that we do not know what will happen if we do not have a second dose. Maybe the vaccine may continue to be efficacious, but what we might have as well is that neutralizing antibody may not be high enough, and we might have also the possibility to have non-neutralizing antibody that may lead to an end of the disease. So this is why I personally against changing any, I would say, schedule in terms of the vaccinations. 2 doses, yes, it can be delayed up to 42 days with the Pfizer vaccine because this is in label, because in the population vaccinated in the clinical trials, there were some with 42 days, but we do not know what to expect if we do not have this in terms of non-neutralizing antibodies. And this is the reason why also we recommend to vaccinate seropositive people, because in terms of safety up to now, we are clean in this group.
Okay, thank you.
Thank you very much. Actually, there are a number of related questions, and I was very pleased to hear that there are ongoing studies in Belgium in cancer patients, because one of the concerns we might have is about cancer patients receiving immune checkpoint inhibitors, which enhance antibody production. Okay, next question.
By Vincent Lenders.
Yes, so you talked about the risk mitigation plan where certain people are excluded from vaccination because of a higher risk to polyethyl glycol, so PEG-induced anaphylaxis. Is that currently already being performed in practice during the vaccine rollout, since non-PEG vaccines are not yet available?
No, what I did say is that they were not excluded, these one, because what was excluded mainly in the Pfizer study was previous, I would say, anaphylaxis with previous vaccines, or anaphylaxis or risks of allergies with one of the contents. And but basically today, what I understand from this reading, also in the SMPC of the product, is that you still have the contraindication in people allergic with one of the components of the vaccines. And one of the components of the 2 vaccines include PEG 2000. And so after discussion also with immunoallergists and so on, it represents only a few part of the population. that would be allergic to, with anaphylaxis allergic to PEG population. And since PEG 2000 is included as a chemical part of some of the lipids, we understood that, and we understand that it is also a contraindication. So the risk minimization plan for this population is mainly the follow-up rather than the contraindication, but we might come with a new contraindication in the future. But to us, it addresses also the PEG, since PEG is in the constituent.
Okay, thank you.
Very clear. We have a question by Tatiana Rivenko.
Yes, hi. I have a question regarding whether the people who receive the vaccine, both doses, do they still have to wear a mask or not? And if yes, why? I don't know if it is true that the vaccine protects only against the complicated cases of COVID and there is no protection for the carriage. Thank you.
I don't know if it's true. What is true is that the data that we have from the clinical trials is, in terms of efficacy, about the diminution, the decrease of the risk of having the disease, the symptoms of the disease associated with a positive PCR, of course, and severe cases. And if you take the Moderna, you have 0 severe cases in the clinical trials with the interim analysis, while we had 30 cases with the placebo. And we take all the disease, then all the symptoms of the disease, of course, it is higher. But there is no data, because the study was not designed to measure the transmission of the virus to others. And so there are studies ongoing, and this is not the company that do that, but this is mainly at different national level for which this indirect efficacy is studied. When it comes, therefore, to the wearing of a mask, the wearing of a mask is, and says also that you see that even after the first dose, you can have for at least 12, I would say, or 14 days, the possibility of to have the risk to get the disease, the risk that you have the transmission of the disease itself. And after, we do not know the risk of the transmission of the disease, but you can have also a transmission by contact. So this is the reason why today, with a low vaccination campaign rolled out in many countries, these wearing a mask and washing hands with hydroalcoholic solution remain needed.
Thank you. Matthias?
Yes, thank you. Well, first of all, thanks a lot for a very interesting talk. Now, I'm an economist, so I will ask a general question about vaccine hesitancy and what we learn about. Now, you did mention the Norwegian case, which I had read in the newspaper, in La Libre, and which worried me a bit about vaccine hesitancy, because in a sense, until now, the story we have heard was that in terms of short-term effect, okay, there were specific risks for very small parts of the population, let's say, and you've detailed some of those. Huh? But here, what we are talking about, according to the health authority in Norway, is that okay, very fragile patients can just basically not take the slight fever or the nausea and die. And they say, we've identified 13 out of the 23 patients where the vaccine was the cause of death. I mean, they are talking. Eh? Now, for the population, I guess the problem a bit is, you know, we've all been told, and I think most people agree with that, that we should vaccinate fragile people, people who are old, who have comorbidities and the like. And these are, but then now the question, the new thing is that, yeah, but they should not be too fragile. So how, to me, it's a bit of a communication challenge to be able to explain that, because, and I don't know whether we can have data on percentage of death and all that. Plus, I've heard also that in Norway, they seem to vaccinate even people who are terminally ill. which may be a bit much. Maybe in Belgium we don't do that. So, but I think I would expect people to be worried about this. So I don't know, what would you tell them to reassure us?
What I can tell them is that, and I could provide you with the estimate in Belgium as well. In the estimate in Belgium, we have every Fridays at this stage today over, I think it's over 380 people who died over a period of 5 days of now in Belgium. So of course we have this background incidence rate, I would say. I think that the concern with this vaccine is that we are focusing, of course, on the COVID-19 vaccine, but the same question could be for the flu vaccine. For the flu vaccine, we also recommend to vaccinate over 65. Okay, so in this case, I would say there will be the first group to be vaccinated in terms of people at risks. What we do not say when they have studied the vaccine is that you need to vaccinate everybody. Okay, and what does it mean by everybody? As I have said, that in missing information, the frail population and very frail population, as it is the case for the flu vaccines, the decision making about the benefit risk of vaccinating this population depends on an interaction with the physician and the patient itself. So I would say that if you want to vaccinate, what is the benefits that you expect to have in terminal patients? I did not make the choice, but in Norway, it seems that some have made the choice and some indeed died. I'm not quite sure that nausea was the cause of death. I know I was a bit surprised also to have that, but we did not see the report as such to know. Fever may be one, indeed, and we know, and I have told you that fever for this vaccine were quite low for both Moderna and Pfizer, except after the second dose. And that, I would say, this may be the most adapted recommendation that we can have today is in very frail patients that have been vaccinated after the first dose and that have suffered from events after the first dose. The question is the same for the second dose. Is the benefit risk estimated positive in the second dose, with the second dose in the very frail patient? So it remains a case-by-case decision making, and we lack data of very frail patients, but this population may be vaccinated, but it remains a decision, an individual decision making.
Okay, well, thank you. And I think this parallel with the flu vaccine, I think could be very reassuring indeed to the general population. Thank you.
Okay, thank you very much. I don't see any other questions, so we are.
Michel, next is Christopher France, who can
Sorry?
ask a question. Christopher France.
Okay, so that will be the very last question, short answer, so we keep the timing. Thank you.
Is that me? Yeah. Do we know already something about the efficacy of the messenger RNA vaccines in relation to the currently circulating variants of the virus? And did this type of vaccine be more prone to resistance than other type of vaccines to new variants?
We have had published the data from the Pfizer vaccines, mainly on the 117 variant, that shows that in lab studies, in immunoneutralization studies with plasma with vaccinated individuals, that the antibodies are still neutralizing these variants. They have performed at least 15 different variants in lab studies before marketing authorization. We have it with Moderna and Pfizer, and also with the following one, AstraZeneca and Janssen, request to have these laboratory studies, but not in clinical trials, of course, because we have the overall efficacy in the clinical trials. But so far, so good for the Pfizer. For the Moderna, we have few data. We have also few data for the old variants, especially not the UK variants, but the other variants. But companies are still doing their study. I'm not sure that they are less prone to do that because they have neutralizing antibodies that are more active on the S1 protein of the virus with different epitopes. But what is quite reassuring with this vaccine to me is that should there be a strong resistance with the vaccination with one of the variants for which we would have a high prevalence in the population, that the advantage of the mRNA vaccine is that they could be modified more rapidly and with a scale up of the production that could be faster than we have with usually used vaccines. So this is a race, of course, now, and I think we need to vaccinate as fast as possible, as much as possible with the good, I would say, vaccination scheduled. But I do not exclude that in the future, one variant will lead to some decrease in efficacy of the vaccine. What we have as a chance is that they have 95% efficacy, these vaccines. So we are starting with a high efficacy, and let me remind you as well that the EMA and the FDA would accept a marketing authorization with a vaccine efficacy of 50% and more. So we have, I would say, some buffer there to have a bit of resistance that could decrease a bit the efficacy of the vaccine. But interestingly, with the mRNA, some possibility to adapt more rapidly.
Thank you.
Thank you very much, Jean-Michel. It's just 2 o'clock, so I would like to thank you very much for your presentation, your clear answer to the question. I would also like to thank all the attendees, and I think that we got very interesting and relevant questions, and I'm sure that we will try to have you back in a few weeks or months from now. Thank you very much, and
thank you.
Nice afternoon to all of you.
Thank you.
Bye-bye.